Hello, listeners, and welcome to the Dr. GPCR podcast, where we bring you conversations with leading experts in GPCR biology. In each episode, we dive into the career trajectories, discoveries, and groundbreaking research shaping the field. The podcast was born at the height of the pandemic with three key goals: to share the latest discoveries, give researchers a platform to showcase their work, and inspire the next generation of scientists to explore this fascinating field. Since our launch, we've recorded over 160 episodes featuring incredible guests like Dr. Brian Roth, Dr. Robert Lefkowitz, Dr. Fiona Marshall, and many more. Our mission is to unite the global GPCR community, encouraging collaboration to improve human health through a deeper understanding of GPCR biology. Thanks for joining us in this exciting journey, and now let's dive into this episode.
Hello, everyone. This is Yamina for Dr. GPCR. I'm very excited to have this morning, and it's morning for both of us, for another 30 minutes for you, to have with us Dr. Dmitry Veprintsev. You and I met last week at the Discovery on Target. What a pleasure to meet you in person, and I'm very excited to have you on today on the podcast. Dmitry, welcome.
Yeah, good morning, everyone, and I'm very happy to be here. I've been following the podcast for a while, and I find it fascinating to learn something about people, something you never heard before. And I think that's important, that scientists are humans at the
Exactly. Thank you for being here. So let's start at the beginning. Could you please introduce yourself? Tell us who you are and what do you do?
I'm a Dmitry Veprintsev. I'm a professor of molecular and cellular pharmacology at the University of Nottingham. My scientific interests are the structural basis of drug action on GPCRs, as you might guess. And the receptors I'm mostly focusing on are cannabinoid receptors. And I've been working on this for nearly 15 years now. And we started off by establishing signaling assays and then profiling a very large number of compounds, trying to figure out what is it that makes ligands agonists or antagonists at cannabinoid receptors. I think it's important to also to know that in addition to being a professor, and I probably can go with later into kind of my background, what brought me to the GPCR world, because it wasn't my PhD wasn't in GPCRs, but now of course we started a small company with one of my colleagues. Providing pharmacological services to the rest of the community, be it a large pharma or a startup. And because the tools we developed are so fantastic, it will be a shame to keep them for ourselves. I think that's the motivation for us to start. So it's called Z7 Biotech. So I think it's quite fascinating that you can combine academic career and being a biotech entrepreneur at the same time. That's what fascinates me today.
So how did you end up in science at all? You mentioned you've been working within the field and developing tools and interested in cannabinoid receptors for the past 15 years. Where were you before?
Well, I think if you really start, right, a lot of values which get imprinted on us by our parents. Right? Quite often, despite the fact we say we have a complete freedom and we know what's good in life and what's bad, the fact is we probably learned this in kindergarten. Or from our parents very early on, and it kind of stays with you, or maybe not. But so I come from scientific family, and the type of books and the type of heroes I would read would be, believe it or not, Cambridge scientists or explorers. So I would know all about Cambridge scientists of like turn of the century, discovering electrons and building liquid nitrogen generators and things like that. And so it's very natural I gravitated towards science. Having said that, it is not really natural that children of scientists don't become not always become scientists, and particularly in the modern world, it's probably not a wise choice, because scientific career is very, very tough, and you can. But nevertheless, that's the motivation. Then, of course, you do have to go to university. So I grew up in a small research center near Moscow. And the research center, it's almost like you think about it as a campus. 7 large research institutes next to each other, isolated from the rest of the world, in a beautiful setting, but very isolated. And most people are scientists or their families. It's a great place to grow up. It's a little bit small when you actually do grow up, because it's only 20,000 people. But the nearest university is in Moscow, and this is where I studied. You do have to compete with others. It's a competition-based entry system, right? So you have to be better than 10 hours, or probably more than that. But at the end of the day, I studied biophysics. And of course, I want to be a marine biologist, and not a biophysicist. Because Jacques-Yves Cousteau, some of you may know, the fantastic documentaries about diving in the most exotic place on Earth was big on my agenda. So my goal was to become a member of staff in his expeditions. But somehow I ended up studying biophysics.
I love it. I do remember as a child, watching his documentaries, and it was a big star when you think about it from a science.
It was a big star. It was wonderful. But what my father arranged for me, he arranged for me to go and see his friend, who was an equivalent of Jacques-Yves Cousteau in the Soviet Union, who was a director of a Marine Biology Institute. And I got to talk to the PhD students. And I was 16 at the time, and they were already 30 and had big beards and families and everything else. But they told me about the reality of life. Said, you only get to go on the exotic expedition once every 5 years, if you're lucky. And that will be in Antarctica. It would not be in the warm seas. It will be somewhere very cold. It is beautiful, but it's cold. So that kind of put me off a little bit from carrying marine.
Yeah, that's what I was going to ask. Did that change your perception on that?
That changed, and biophysics was the second choice, because I've read all about the golden age of biophysics in Cambridge and after the World War II, and that was fascinating, like ion channels and.
From the Soviet Union, you mentioned that you went to university at the University of Moscow. How did you end up in Nottingham? How did your path lead from biophysics to GPCRs today?
Well, imagine you go on holidays and you come back, and the country you grew up in doesn't exist anymore. As simple as that, and you have tanks on streets. So that's what happened when I was finishing university. And when I started university, because of the Iron Curtain, there wouldn't be a chance to go abroad, because for most people. So you basically, despite the fact you may read the books exciting about scientists somewhere else, but you know that's not going to be your path. But then it became the possibility. So I ended up doing a PhD, which involved going to the United States. And I did my PhD in protein folding field. So protein biophysics. And the most famous person in protein folding field at the time was Alan First in Cambridge. And somehow I really dreamt that I can go and do a postdoc with Alan. So I ended up emailing him and saying, can I come for a postdoc? I will get fellowship. What could be easier? And come. So somehow I managed to get a Human Frontier Fellowship by hook or by crook. And come to Cambridge for a couple of years, and I ended up spending 11 years with Alan First and Greg Winter in Cambridge. Focusing maybe not even on protein folding anymore, because also his interests have changed by the time I got to his lab. So I worked in cancer research. I worked on a protein called P53, which is one of the major tumor suppressors. And at the time, the problem we had was to understand how does it work. What does it look like and how does it work? And of course, the second question was, we have a lot of mutations which cause cancer. Is there anything we as biophysicists can do? And we came up with a concept of chemical chaperones, like small. And today it's quite common, right? But the idea was, have a small molecule which binds this destabilized mutant, because most of them were just destabilized. So that was an interesting outcome of biophysical work. So if you can stabilize it, you can restore its function. That was the concept. And I think after many years, Alan succeeded to develop in one small compound which stabilized a particular mutant of P53, which is quite remarkable. So drug discovery is tough. But of course, by that time, you cannot be a postdoc forever. I became a staff scientist in the lab, but then I really had to find my own identity. And membrane proteins, we used to call them a career suicide. So whoever touches membrane proteins, that was it, right? You'll never see this person again.
Well, the whole podcast is about one type of membrane proteins, and yet we're here.
But it changed. That was a realization that by the time I was kind of really desperate to become a group leader somewhere. What has changed that we could express GPCRs recombinantly. We can purify them, and we can do some biophysics with it or structural biology. So that was the time when the first structures were just coming out, right? And when one of my colleagues from Cambridge got a position in Switzerland, Gebhard Schettler, so he's was one of the structural biologist pioneers in structures of GPCRs. He was looking for a bunch of group leaders to join him. Okay. Because he became a head of a division. And that was my opportunity. So I went to Switzerland and focused my research on GPCRs. I talked to Michel Bouvier, said, Michel, what is the best receptor I can study? And he gave me tips. One of them was vasopressin, and the other one was cannabinoid receptors. And at the time, we had this illusion that we can make GPCRs in E. coli and label them for NMR studies. Now, 15 years later, I think I already know the answer to that. But A, it's very hard to make GPCRs in E. coli. Yeah. And B, even if you manage to label them for NMR, there is very little you can learn because of the intricacies of the protein NMR and how GPCRs look. Yeah. But we have succeeded in this, but not labeling every amino acid, but just individual amino acids. So there is a lot you can learn about GPCRs by labeling some amino acids in your protein.
Absolutely.
So that was the beginning. And then, of course, as you always do, you write your grants, you get your students, and then it starts. And then, of course, working with GPCRs is incredibly challenging. Yeah. So it really drove the development of a lot of technologies we use today. Like, how do you study a protein you cannot even express in sizable amounts? How do you study a protein that denatures before your eyes once you purify it? Yeah. So all these challenges are quite fun to overcome, but it just takes a long time.
It does. And you mentioned growing up in the Soviet Union and not even thinking that that's going to be possible to use. I was born in Romania, and I lived a little bit of time in Romania before '89, obviously. And even at that time, it was this difficulty. You couldn't leave the country. You couldn't get things in. I do remember that by the time my family left, we used to go back, and you had no coffee. We were bringing in coffee, even lemons, back in Romania. So I know, I still remember those things where it was complicated. But then times change, and I feel like opening the doors and being able to travel abroad for a scientist is such an enriching experience.
Well, I think that's one of the pleasures of science, that you can go anywhere in the world and find people you can talk to, very similar people, whatever they are, and the pipettes are the same. Yeah. And often, even the language is often the same, because it's often borrowed from either German or English in the first place.
Exactly.
So it is very easy for scientists to actually be very international, and I do enjoy that, I have to say. It does come at a price, right? Your childhood friends are scattered around the world, but they have their own careers, so they also travel. Right? So
exactly, and then you meet if they're scientists, you meet up at conferences.
If you're scientists, you meet at conferences, but of course, sometimes you make good friends when you're older, but often it's the best friends are still from your childhood.
I agree. No, I absolutely agree. And then, so you're in Switzerland. And then how do you end up at the University of Nottingham?
Well, so in Nottingham, so some of you may know Steve Hill, and he was very, very successful at organizing the Center for Membrane Protein and Receptors. So he united about 30 research groups working on membrane proteins and receptors. mostly GPCRs and RTKs, but also some other membrane proteins which are important for blood clotting. And then convinced the two universities of Nottingham and Birmingham to fund this for 5 years. And within this money, they had opportunity to hire 2 professors, and I was one of them, or 3 professors. So that was quite remarkable. I have to say that is a tremendous initiative, because you really want to be in the environment where you have a lot of people to talk to.
Yes.
And fluorescent ligands were developed in Nottingham, and I think, okay, what is the true, unique development coming from Nottingham? That would be fluorescent ligand detection system. Right? That's what replaced radioligand binding assays today. And of course, all the signaling and classical pharmacology. I'm not a pharmacologist. I'm a biophysicist and structural biologist turned pharmacologist. So for me, it was a tremendous learning exercise as well. Starting to teach on pharmacology courses a little bit, and or pharmacy courses, and or medical students. My previous career was always in research institutes. So I did my PhD in the research institute. I did my extensive postdoc and LMB in Cambridge, a research institute. So the younger students you see are PhD students, or occasional master student, or summer student. But you're never involved in teaching. At Switzerland, again, I was at the synchrotron site, Paul Scherrer Institute. Again, I do have occasional master students. I did teach at ETH, just for the purpose. Students don't know you exist until you teach them. That's a very interesting observation. You would think how hard it is to find anyone in the world modern days. It's apparently impossible. It's just you don't exist unless you're a professor, and they have to pass the exam. Suddenly become very important.
Exactly, and then they want to work in your lab.
This was an opportunity, and and of course in the academic path, that's kind of become professor is probably holy grail for most of us. And also all the intermediate positions disappeared. I might have been happy to be a research scientist, but these positions just don't exist anymore. So if you want to stay in academia, the only path is to become professor.
Yeah.
Otherwise, go away. It's a very brutal world today, right?
It is. I was going to ask you, but I think you alluded to it in the beginning, is why academia and not industry? And you're right now, you're a little bit, you have your foot in each. But back in the day when you were doing your postdoc, have you ever thought at the time, okay, do I want to be a professor, or do I want to find a job at a company?
I honestly, retrospectively think it's pure ignorance. I did not know how exciting the career in industry could be, and I certainly didn't know what entrepreneurship is all about. That is crazy how little education we receive. A school and university prepare us to be good employees, believe it or not.
Yeah.
So go study well, get good grades. Don't get out of a line, not too much. Be a good boy or a good girl. Be a good employee. Do what you're told. That is not helpful. Academic world, of course, offered this freedom. In the countries like Soviet Union, academic world was probably the only place where people could hide from the system, because nobody really understands what you're doing. So that was a place where a lot of talent was hiding. And the moment the system collapsed, a lot of them became entrepreneurs, because that's what they truly wanted and didn't have an opportunity before, and now they had it. And now they did.
Yeah.
And that's very interesting. I mean, I had, even if you haven't inclinations to be an entrepreneur, if you don't have a right ecosystem around you, you don't know the people who've done it before, it's impossible. You don't even know how to approach this problem.
I agree, and I think universities don't prepare students, including me, for leadership, because even though maybe you don't want to be an entrepreneur, maybe you don't want to have your own company, but you want to lead a successful group, you kind of learn on the go once you become a professor.
True.
But to your point, entrepreneurship, building a team, building something from the ground up is not taught. So if you want to do it, it's on you.
It's on you. And I think most of people who've survived and achieved building their first group, right? And go walking into the empty room with whatever little startup money you have or don't have. I mean, I've been through both. I've been starting with zero budget and starting with a sizable chunk of money. It is still very challenging. How do you? You're alone. You're a group leader of yourself.
Exactly.
And when you get your first master's student or Erasmus student, in Europe we have summer students called Erasmus students, so they come from abroad, and typically they're the best students you can get because they jumped over the hoop of going abroad, so that puts them at top 10% already. And this is your group. And when it takes a year to get your first PhD student, it takes another year to train them to become productive, to learn the techniques. So it's a very slow process, and of course you have to keep writing grants, and it's not dissimilar from being a startup. Basically, either you pitch to VCs or you pitch to the granting agencies.
Exactly.
Maybe the difference is there are many more VCs than granting agencies.
Yes, and the granting agencies don't want necessarily their money back. Then
The granting agencies don't want their money back, but you can only apply for them like once every 6 months. So, small probability of that. So the granting agencies do want their money back, and other types. They want their money back in papers. They want to make money in progress and PhDs.
But it's different.
Who become professors on the long term.
No, absolutely, you're right. But it takes longer, so they're not going to come back to your door a year or 2 years later saying, "Where's my money? Did you, double it, triple it, or did you make tenfold of it?" Compared to VC, so it's a different type of reward there. Then let's dive a little bit into the GPCRs aspect. You mentioned that biophysics and GPCRs happen to be good, example proteins to study biophysics, and that's how you kind of ended up in there. And you also mentioned that Michelle. So let's take a moment to say that Michelle is a wonderful mentor. I owe my interest in GPCRs to Michelle because I was in his. And I've said, for those who listen to the podcast regularly, I explained this multiple times. I was in his undergraduate class and we were talking about signaling. It was all about signaling, and he was teaching GPCRs. And that, to me, that was fascinating. How can one protein collect the information from outside and then get the information inside and then the cell to respond? And he's been an incredible mentor since then. When he suggested vasopressin versus cannabinoid, what were the arguments against vasopressin in your case and for cannabinoid? How did you end up working on cannabinoid receptors?
Well, as I said, I think I met Michelle with probably in the first month I started my job in Switzerland, and he was visiting Switzerland conveniently, so we had dinner. I think he was visiting Gebhardt, of course, and we were just chatting. And of course, for me, applying biophysics, and I'm new in the field, right? You have to realize, I knew nothing about GPCRs. I'm completely naive. There are 400 receptors to choose from. Which one do you choose? And I say, okay, what is the best studied receptor? Where do we understand pharmacology? We have enough tool compounds. It's very much like, what is it a good target for biophysical studies? Unfortunately, at the time, we didn't really understand what makes a good target for biophysical studies. So vasopressin was not a good target. It was a very challenging target. And of course, we tried to get structure of it. And the groups in Montpellier succeeded eventually after probably 20 years stint of trying to get structure. So it was incredibly challenging. And so did we. We tried, we did crystallography and, produced milligrams amount of vasopressin and set up endless plates, and none of the crystals diffracted. But that's kind of the nature of being a structural biologist. That's, if you're not lucky, you don't get diffraction. And sometimes if you're lucky, then you get it. So. But despite of that, that was a starting point. And once you start making vasopressin and receptor and finally established ligand binding assays, finally established signaling assays. You kind of, I wouldn't say called trapped in it, but and it's a good system. It activates every G protein under the sun. So it actually, for a lot of biophysical work we did, it was a good model system. We ended up pivoting our research away from like traditional biophysics, where you purify protein, you purify proteins, and moving into what I call today in vivo biophysics, which is like in cellular biophysics, where you don't purify your proteins anymore. And what you do, you develop assays which are so selective you don't see the rest of a cell. You don't say, oh, we have 10,000 proteins which are there. You only focus on one process only, and you can do it with great sensitivity. And that made the whole difference. So now you can introduce mutations in your receptor and see how they affect the signaling and quantify this. You can quantify the effects of ligands on signaling. And that makes all the difference, right? Being able to do that systematically. Vasopressin, of course, was not a great system because it doesn't have a lot of ligands. It has a handful. And it was a hot target back in the day, but it was out of fashion. And cannabinoid receptors, have been a hot target for a long time, 9,000 years precisely. So it's probably one of the first receptors targeted in traditional medicine. And of course, CB1 and CB2 have been targeted by massive drug discovery campaigns by multiple pharma. So I have a lot of ligands in the literature. And there was a group at NIH which expressed it in E. coli. So we thought we could do that, repeat their success, and label it for NMR, because that's, at least coming from a soluble protein field, you always label your proteins in E. coli because you can do uniform isotopic labeling. So that was the goal, and that was not what it was not, or is still not possible in mammalian cells.
How cool is
And then we spent years trying to express both vasopressin and cannabinoid receptors in E. coli, and we failed miserably. So we made milligrams of unfolded material.
Wow. I always ask this. So if you had to pick your favorite GPCR, what would that be? Cannabinoid or vasopressin or any other GPCR?
Well, I must admit, we use beta-2 adrenergic receptor as our guinea pig, as everybody else does. It's a wonderful receptor. Like it is really, really good, and there is still plenty of new ligands to develop for this receptor. And we're discovering new things about it, despite this being one of the first receptor being cloned, and the first receptor for which drugs were developed, beta blockers, by who developed beta blockers?
That's a good question. I'm blanking here. Yet it's
James
still
Black.
Yes.
Yeah, by James Black. So we have drugs for it for 50 years. And of course, propranolol is probably one of the best-selling drugs in the history of medicine, right? So still.
Yeah, no, I agree. And it's interesting because it always works. The beta-2 has always been
it's a wonderful receptor. So we use it as a go-to system. Every time we develop a new assay, beta-2 would be the first one we try it on. But of course, and also, whenever we develop machine learning approaches to understand what makes agonists, we also did it on beta-2, just because of the sheer amount of data we have for it in the literature.
Exactly.
Cannabinoid receptors, and of course, it's a good target. Cannabinoid receptors are fascinating. They're quite challenging. Of course, maybe I'm biased because I've invested so much time working on them. They're there and they're not there, and what exactly do we do? We still don't quite understand. So we have wonderful drugs today in the clinical trials. So I think that large pharma and medchem people did wonderful. They're very selective, bioavailable, soluble. They are fantastic drugs. They don't get degraded by the liver. Everything you could dream of as a drug, and yet they fail in the clinical trials. And the question is why. And when you start really thinking broader, so if biophysics tend to be focused, or molecular biophysics tend to be focused on one protein at a time, and sometimes two proteins at a time, like protein, your receptor interaction with a G protein, or your receptor interaction with arrestin. And that's kind of where biophysics stops. And then you start asking the question, okay, why do the drugs don't work? And that is a big challenge.
It is. No, I agree. I think that.
So and then, well, we have some ideas.
Yeah, I recorded a podcast episode yesterday with a postdoc currently at Stanford University, and we were talking. So Gabriele's PhD was all around microscopy and trying to see how the membrane composition affects GPCR activity. And so we were just talking around the fact that even physically, let's say squishing the cells or the cells getting ready for chemotaxis, how it changes where the receptors are. And in his work, he was able to see that depending on where the receptors are in the membrane, an agonist activates differently. Some of these receptors that are in one group have 20% higher activity, constitutive activity, than the receptors, and at a different density in the same cell at different position, and then adding the ligand changes. So I think, to your point, biophysics is fantastic. You get a sense, but it doesn't paint the whole picture. And this is we were just talking about the cell. Can you imagine about the whole organism?
Yeah. Well, I mean, that's fascinating. I'm actually I probably have to look up this person and figure out how they do it, because that sounds tremendously challenging.
Yes, I will.
And yet it does make a lot of sense. Yeah.
I will put you in touch with Gabriele. He was fantastic.
Yeah.
Very, very nice work.
So I think one idea that fascinates me in this direction is a combinatorial action of hormones or drugs. Yeah. And the idea has been very simple, that actually you probably need to simultaneously engage multiple receptors with your drug or combination of drugs, because that's how our body communicates. Yeah. It is very rarely that our body sends a single hormone to do its job. Yeah. So it's quite often a combination of hormones, and that's very much like postcode in our body. So if you have a combination of several receptors, that is far more likely to be unique for a given cell, rather than one single receptor, which is expressed, probably in half of the cells in our body. So whatever be that adrenergic receptor, if we take this as an example, we say, it's a good target in the heart, but it's expressed everywhere. If I throw an agonist on beta 2, I may restart the heart, but I also would activate a lot of other stress responses in the body elsewhere. So if I want to be very selective, I may want to activate beta 2 and something else, which in combination only exists in the heart.
Yeah.
And maybe do it with a very weak partial agonist, so they actually don't trigger a massive response elsewhere by themselves.
Absolutely.
And they only trigger the response in combination. So that's kind of my current area of research, which I'm exploring. And of course, that automatically lends itself to new ideas for the companies, right?
Exactly.
It is an academic exercise, but actually it is a drug discovery exercise.
Yeah, it is, because you need to understand what happens in the system, and then how do you relate whatever happens in the system to a disease. And then if you modulate it, you still have to understand what you're doing from a receptor perspective anyway. So tell me a little bit more about where did the idea of starting a biotech company or a CRO come into the path of working in biophysics?
Well, think about like these random events in your life. Completely random, right? So I'm a struggling PhD student, right? I did my PhD part-time United States, part-time Russian Academy of Sciences. My fellowship at the time was $50 a month. And the inflation was horrendous. So this $50 lasted about less than a week. So how do you supplement your income? I was good with computers, right? I'm one of the first people who had like websites, like the first 100 people with a web server. Mm-hmm. The early days of Linux. And I got a job offer from a bank. Say, come and become our chief IT security, because people who knew something about the internet at the time, they just didn't exist. And they offered me a salary 100 times more than I was making.
Wow.
100 times. And I looked at it and said, it was very tempting, right? I am poor. But it just didn't align with my values.
Mm-hmm.
I'm really not interested in guarding somebody else's money.
Yeah.
I mean, that's what chief IT in the bank does.
Absolutely.
Right? You're guarding somebody else's money from internet thieves, right? So it doesn't align me. I don't care about somebody else's money. What I care is, it turns out that I have my values, which I'm probably imprinted in my childhood, I like to build stuff, and I like to explore new territories. And it turns out I also like freedom. So, it's very hard to be part of a big system where people tell you what to do.
Yes. Yes.
So when you make this decision, say, all right, well, actually, I'm going to get my PhD in a year or two. I am going abroad for a postdoc by hook or by crook. So I don't need to take this job. I probably just have to persevere and get my PhD done, even if I'm poor.
Mm-hmm.
But at the time, being in the United States, I used to call back home because I was homesick. That was very expensive. $1 a minute. And as a poor student, you cannot afford that. So I had a brilliant idea. I can call from the internet. I have a computer on one side and another computer on the other end. And I knew how to build it. So I went to somebody who had enough money to sponsor 2 computers. I said, I have a brilliant idea. I can make telephone calls over the internet. That will cost nothing, right? And people are paying $1 a minute.
Yeah.
And I don't have this. I mean, who has this problem? And these people were rich. They didn't mind spending $1 a minute calling. And they were not in Boston either, right? So
yeah. Well, I can tell you that while I was growing up in Algeria, my mom, we couldn't call Romania. There's no such thing as you can pick up the phone. So we had to go to the post office to write and send a telegram.
Yeah.
And when I think about it now, just saying it out loud, I sound very old, but no, this wasn't a very long time ago, where you couldn't just pick up the phone and make a phone call.
Yeah, exactly. But that was kind of the first brush of entrepreneurship, right? Okay, I failed miserably at the first obstacle.
Yes.
I was 25. I looked like a 16-year-old. So obviously, I didn't make any impression of these people who could have invested in me at the time. But it was way before Skype. It was way before these telephone scratch cards appeared, where you can call for 1 pencil or 1 cent a minute, and you earn the world. So that was like a few years before that.
Yeah.
Of course, everybody had the same idea, because if I came up with it, I'm pretty sure a lot of clever people came up with the same idea.
Yeah.
And then you think, okay, what way do you continue your postdoc career, and think, and you see remarkable things. You see people starting companies next to you. So Cambridge was great.
Yeah.
I saw several companies being born in the corner of a lab. And then suddenly, former PhD students becomes the first employee. And after a couple of years, they have to move out because they outgrow. Suddenly, they have 2 or 3, and then they need a whole bench rather than a corner of a bench.
Wow.
And then that's the end of it, right? They have to move out. But these companies then develop, and that I saw the birth of Diamantis, single domain antibodies, which were eventually bought by GSK.
Yeah.
I saw the birth of Bicycle Therapeutics, where actually was one of our own PhD students was their first employee. I saw the birth of Heptares in a very similar way.
Yes.
And you think, there's a lot of value in what we do.
Yeah.
And you see people actually enjoying it. And most important, they actually move to the company. Forget postdoc. We go and build something. So that was very exciting. Of course, at the time, you really needed starting a company, starting budget was incredibly hard. So it was always based about the reputation of people, of the founders.
Yeah.
Today, starting a company is incredibly easy. It's much easier compared to 20 years ago.
Absolutely.
So you don't need the same amount of money. You don't need, and also the connections are there. LinkedIn is a wonderful tool. And of course, I'm in a very different situation. I already have a lot of connections. 20 years ago, I didn't. I wouldn't be able to come up to anybody on a conference. I was so shy. I would go to the conference and hide behind the posters.
So let's stop and talk about that a little bit, because I think you were not there. There's other people who feel the same way. How did you overcome that?
Well, I didn't. I'm still very introverted and very shy.
You can't tell, by the way. I couldn't tell last week when we met in person. I can't tell now. So how did you work with your introvertness in order to still allow you to talk to people and network with people and develop those relationships?
Well, I think, the number one was the realization that it doesn't matter how good the science you do on the bench. If you don't tell people what you do, nobody would know ever.
Yeah.
They do read papers occasionally, but very few of them. So it is strange, right? But even people in my own group haven't read all the papers I've published. I say, well, we've done this before, right? In this paper, I say, oh, right, okay, that's good. How I read it, and it's fine. It's not like everybody has to study all the papers I've written. It's not all of them are good. But it's really important to realize, actually, first of all, other people are just as shy and introverted as you are. Like most scientists are introverted. That's what makes them good scientists, because they have time to think on their own. They also desperate because they're also very interesting people. So, and just on a human level, they're just good people, right? Science is a slice of a population. You would find people with the same hobbies you have, same likes and dislikes, same ideas or different ideas, which is even more interesting.
Exactly.
So it basically out of a necessity. I mean, you go to the different courses and you say, well, networking is really important. You read all the wonderful books which says networking is really important.
Yeah.
When you realize networking is really important, actually, it is really important. So, I was not taught this as a student, right? Our presentation skills just didn't exist, like giving public talks. Oh.
Yeah. No, I agree. And
when you just start doing that, and actually, it's not that scary, right? As scared as I am.
I agree. I think it's important to stop and highlight the fact that we're all human. And the word networking, I think it's overrated, because it makes you think that it's this big deal when it's just talking to people.
No, absolutely. I do what you do. I really like to get to the background of people. Actually interested in the person, right? I really want to know, where did they go to school? Which university? What motivated them to do this and that? How they end up in science in the first place? I guess you get people from all walks of life. I think, how did you end up being a scientist? What motivated you to do it, and why you're here?
No, absolutely. And that's when you build a bond with the person.
And very often it's just chats, right? I don't really need anything from this person.
Exactly.
I'm just interested in them. Like, I see their poster or I see their talk and I go, that's cool or fascinating, or it's really, really great work. And but at the end of the day, I'm more interested in personalities, what's behind that. And sometimes that translates in collaborations.
Yeah.
In the biotech world, sometimes it may result in biotech collaborations, because you might be trying to solve the same problem and you have different parts of a puzzle. But it's really important. I mean, it's actually knowing what other people, what they do and where they are, and putting yourself on their radar somehow. That makes all the difference, because you really need this collection of friends almost. You can email and says, "Oh, I have a question. You may know the answer."
Exactly. And I think it allows also people to. I mean, I enjoy helping people. If I can introduce you to someone you might want to talk to, and it forms a good relationship, a collaboration, you name it, that makes me feel good, because I was able to put those people together. And that's the whole point with Dr. GPCR as well. The podcast is to really have this type of conversations, but to inspire junior scientists. But the whole concept is around bringing people together, because I think science is so difficult. GPCRs are not a walk in the park, although we all love each of our receptors and etc. But being able to connect together and meet and have conversations and exchange on our science, I think it's very important, because otherwise we would all be working in our corners and not communicate, and most likely trying to facing the same problems no matter what. So absolutely. So tell me a little bit more about Z7 Biotech. What's the inception? I know a little bit about it because we talked about it in person last week, but this is more for the audience. And you kind of hinted to it when you were describing, growing, starting a company in the corner of a lab and going from one employee to two, and then having to move out. But I'm curious to see, where do you see the company in the next 3 to 5 years?
I mean, it sounds. I think it's the most kind of a challenging things I had to learn in the last 3-4 years since we started the company is about entrepreneurship and how do you grow the company. And that actually, for me personally, that was a huge learning experience, and I also developed as a person as a result of it, and probably changed a bit. And it all started in COVID times, right? My colleague who is in my group, he's David Sykes. He's ex-Novartis, so he's been a drug discoverer all his career. When Novartis decided to close down the UK site, and scientists got scattered, as it always happens, right? Some moved abroad to join other Novartis sites, some stayed in the UK and then have to find a place. And David ended up at the University of Nottingham and in my group.
Mm-hmm.
But I knew him from before. That's the power of networking. I met him on a conference in Switzerland. He was presenting a kinetic profiling of compounds. I talked to him. I really wanted him to join my group in Switzerland. And by twist of a fate, I ended up moving to Nottingham, where he was at the time, on the same floor.
Wow.
So I convinced him to join my group. And
what a small world.
And then in COVID times, I was sitting there. I said, well, we have all the skills. We can discover drugs instead of sitting there and doing fumbling out, whatever we did. So we started a company. And COVID times was wonderful because you can Zoom and talk to everyone you wanted. So we've talked to about 100 people.
Mm-hmm.
And both of us had enormous networks in industry. So we talked to pretty much everybody who does drug discovery in big pharma and ask them, what's your problem? Yeah, very simple question, right? What is it you have to deliver for the company, and what is your burning problem?
Exactly.
And then we combine it with our expertise. I say, well, actually, we're very good at profiling GPCRs. And that's what we ended up doing. We developed a company so profiling ligands for others, because we can do that better than many others. And that is as simple as that. It has evolved a little bit since then, and we expand our repertoire of assays and what we can do, and move them to. But I think that is kind of the gist of it. It's very simple. We just develop better tools to profile ligand engagement and ligand signaling, and that's fascinating because you actually get to see with very exciting projects. It stretches you as a new target you never touched before. You have to develop the tools now, fast. So that means we develop things which are scalable. So you can throw a new target at us and it will come up with an assay very quickly. So that was fascinating. And of course, also very, very reward, right? I wanted to build stuff, and that's something you don't get to do in academia very often. And now I'm much closer to the action of actually developing new drugs. I'm helping companies to advance their candidates to the next step of a drug discovery pipeline.
That's a great point that you're making. You know what made me when you mentioned this about learning new targets. I work with a lot of CROs, and oftentimes when you think about a CRO, you're thinking about a tool that you use to get data. But I think on the other end, when you're a CRO, you get the opportunity to develop new things, and you acquire a collective new knowledge that you can then apply to different projects. So it's not only beneficial for the biotech or the pharma company, but it's also beneficial for the CRO and for the tools and services they offer.
Yeah, it trickles back into our academic research. So
yes,
some of the tools we developed are so superior that we start using on our academic life as well.
Yeah.
And it makes a lot of sense. And of course, for young people, I think it's important to remember that even in the company, I'm on the bench, maybe occasionally, but not very often. anymore,
Mhm.
but I
Mhm.
can roll up my sleeves and I can with a pipette and get results. Often these are young people who are on the bench and they're developing their own careers and their own. Yes, for all of us, right? We have to be motivated to do what we do. And
Yeah.
if in the past, the students would walk into my office or my lab and ask you, I would test them on their technical skills, right? Can you solve the equation for ligand binding for me in 5 minutes, please? Today, I'm asking them, what's your dream?
Yes. No, I agree. That comes with
So,
experience as well.
So what do you want to achieve? And because what you are here, that's like a stepping stone in your career.
Yeah.
So same for the company. So do we see it's the company developing in the best GPCR profiling company? Yes, for sure. That's the ambition. But it's also a stepping stone for us ourselves and for the people we employ. For our first employees, definitely is a stepping stone. What's next for you? You're not going to be a bench scientist forever. Who would you like to be?
Exactly. And that's a very tough question, I think, to ask, because it requires thinking, and it's not a yes or no. It requires over and over thinking about, okay, what do you want to do? What are you good at? What are the skills you want to develop, and how much time do you want to spend doing that in order to reach your goals? There's lots of
No, absolutely. But if you think about it, you have no choice. You have to do it.
Yeah.
If you have a satisfying life, if you want to have a satisfying life,
yeah,
it has to be aligned with your values, and that means what you do has to be aligned with your values.
Yeah.
And I unfortunately see a lot of people who do something they don't really like, which doesn't bring many joy in a sense of life. But yeah, it's a job which pays their bills, and it's not. I mean, these jobs have to be done, but some of them could be automated because they're not great, and some people may find an alternative career very much happier.
It is,
and some people they may be very happy doing exactly what they're doing. That's the thing.
Exactly.
It's maybe a dreadful job for my eyes, but it's a dream job in their eyes, and that's fine.
Exactly. No, to your point. I think you make a great point there. I think it's easy to say, oh, you should do what you like to do and find your dream job. It is harder to get there because sometimes life comes and knocks and says you need to do this, but at the same time, not losing the objective. You might be doing something right now to pay the bills, but as soon as possible, you can move on and you can prepare that. And to your point, there's a job.
Crazy things to pay my bills as a student.
me too. Me too.
I've been a taxi driver. I don't want to be a taxi driver again.
I used to deliver newspapers.
All right.
From 3 to 6:30 in the morning, and then go to the lab afterwards. And I actually even fallen asleep going home on the train and missed my station and had to go back. But it's the price you pay. All right.
So we have to do it, right? You do what you do. Yes. But as long as you're absolutely right, as long as you remember you have a goal, then you can make choices which brings you a little bit closer to that goal.
Exactly.
Maybe if I still be minimum wage, but at least it's a step in the right direction.
Exactly.
So over time it compounds.
It does. And that's one of the goals of the podcast as well, because when I think about today's PhD students or postdocs, and they look at people like you or people who have been in the field for 20-30 years and they published abundantly, and the first idea you get is, oh, that person just happened to be so lucky, they've always gotten all the results, they've published, and they're set, and they didn't go any through difficulties. But being able to talk about these topics and actually explain that no, you had to fight for everything that you have, and you had to think about what your goals are, and you had difficulties, having issues with the receptor, that you don't have the right tools to study, or being a taxi driver or distributing newspaper. I mean, I had 300 plus clients. And on my last day, I left each of them a little card saying, I've completed my PhD. I will no longer be delivering your newspaper. And I got people who called me and he said, I didn't even know that, the delivery person has a PhD or is going to have a PhD now.
Yeah.
And you get to meet all sorts of people and you learn from that. You learn to value what, you learn to appreciate what you work towards. And when you reach your goal, then you realize if you're anything like me, you realize, okay, I want more. I need more. I get to this step and then what's next? And that's important also. I'm conscious. I'm just going to say I'm conscious of the time. So I have 3 more questions.
Yeah.
Top 3 aha moments that you had as a scientist that shaped your trajectory. And I'm going to take notes.
A very quick question. When I was going to university, and it is very unfortunate, but I joined the Department of Biophysics, which had its heyday in the 50s and 60s and 70s.
Mm-hmm.
And that was the days where the technique was king. So learn the technique and you'll be good forever. And that kind of, it really left an imprint on me that, do your job well and it's fine, right? The rest of the career would fall into place. And then I come to Cambridge, and even in my department, other groups would say, ask the right question. Question is the key, ask the right question. But that's something which I had to relearn or learn again the hard way in Cambridge. And that was a great place to be in Cambridge because people around you say, ask the right question. And that is the aha moment. When it sinks eventually, you say, no, it's not good enough to be an expert in technique X.
Yeah.
Whatever it is you do, you really have to ask the good biological question. That completely changed my trajectory. When I started thinking, hey, what is the good biological question? How do you even ask one? But the moment you start thinking about it, so that's number 1. So I think the realization, the networking is number 2. The fact we can actually talk to other people, will talk, and most of them will answer.
Yeah.
That was crazy thing, right? They actually, they're normal human beings like I am. They are not gods.
Yes.
They also have something trivialities, dogs and cats and children and whatever.
They have to eat, sleep,
more
exercise.
loans to more. So they're normal human beings. That's number 2. And number 3, I think we have the whole idea that actually science is great. You have enormous freedom. And you can really start changing the things you're working on.
Yeah.
If you ask the right question and you say, put a bit of effort, things will start falling into place over time, not immediately.
Yeah.
But with time, things will fall into place and you can start your company, you can do whatever you want.
No, I agree.
It was really important to start understanding yourself, like, what is it I want? Instead of just saying, oh, I need to make a career. Who is successful here? This person. Can I emulate their career? Well, that doesn't sound quite plausible.
Yeah. No, I agree.
So if you there's a quick answers.
I like it a lot. I was taking notes because we're going to be making little clips of these. And so the team needed also a title for the little clip. So thank you for that. All right. Any advice for young scientists who aspire to contribute to the field?
Well, I think I already did. So that's the first question I asked them. What's your dream? And I think this has still been my first advice to the people, actually. When you start in the field, think about it. What is it your dream? What do you want to be in 10, 15, 20 years? And don't be shy. Think that everything would pan out.
Yeah.
Because, whatever it is, you may want to be a professor in famous university, or you might want to discover a new mechanism of action of something and be known for that. Or you may want to start your own biotech, or you may want to be the best lecturer under the sun.
Yeah.
And teach on Coursera. You may want to deliver a TED Talk. That's my dream.
A TED Talk? Yeah. I mean, I watch a lot of TED Talks, and I think it's fascinating how how communication actually has an impact. If you can be the best scientist in the world at what you do, but if you cannot communicate it to your colleagues, to the general public at a high level, then it's like there's a disconnect. You'll never be able to inspire others and communicate and also help move any field forward. So I very much agree with that. I like that one a lot. Very much. All right, last question, which is I think the toughest one. What should we title this episode?
Oof.
Yeah, I know. This is the reaction of every guest. I started using that earlier this month or last month, and it's a tough one. I know it's a tough one, but I think what I just captured for the top 3 aha moments could be something interesting. You know, I captured ask the right biological question, talk to others, and they will answer. That's what I took note of.
Yeah.
I think one of these two.
One of these two. I mean, the third one was about your values.
Yes.
Right? Know your values or know your dreams. It's the same thing, to be honest.
Yeah.
But
yeah. So as a catchy title, I think it can be I ask the right GPCR question, or I think talk to others and they will answer. I think this is a little bit more cryptic.
A bit more cryptic. Well, I would go with any of them, but
all right, then we're gonna go with the first one. Ask the right GPCR question.
Ask the right GPCR.
Yeah. Oh God. Okay. I need to. It's a different keyboard, so I'm doing stuff that I'm not supposed to do. 1 second. I have it. Oh God. Okay. All right, and I'm gonna write this down. Ask the right GPCR question. Dmitry, thank you so much for your time.
Thank you.
I really enjoyed our chat.
Yeah, it's been an hour.
Yes, you know, it
no, I thought I was aiming for 40 minutes, I'll be honest.
No, this is what happens. I really enjoy doing this, and sometimes people come in on the calls and say, oh, we're going to do this in 30 minutes and we're done, and we always end up talking more. And that's kind of, in a way, my superpower, because we get to talk and we get to know each other.
Yeah, but you're very experienced, and I have to say that you're very smooth, so it's easy to talk to you, so I think that is the good thing.
Thank you so much. Really appreciate it. This was episode 163 that we just recorded of the podcast. All right. Thank you so much. Don't go anywhere.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. 1 click goes a long way. All the information about Dr. GPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the Dr. GPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email Yamina at drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members: Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. Until next time, and stay safe.